Welcome back to the deep dive. Today, we are wrestling with, well, perhaps the most profound biological contradiction, really, in the human body. We have this immune system. It's aggressive, powerful, this specialized army designed solely to recognize and destroy anything foreign, anything that isn't us.
It is truly a remarkable piece of biological engineering. I mean, when this system spots a virus particle or maybe a bit of bacteria, even just a foreign protein, it just launches this huge response. You've got T cells, B cells, these complex chemical cascades. Everything is tuned for elimination, and with extreme prejudice, you could say. It's an incredibly successful defense strategy.
But, okay, that success immediately brings up this massive, fundamental question, right? If this defense system is so ruthlessly efficient at identifying and killing off foreign invaders, how on earth does it manage to coexist peacefully with the trillions of cells that make up you? Your skin, your liver, your brain, your heart. Why doesn't the army just turn on the homeland?
Ah, yes. That ability, that critical skill to differentiate friend from foe, to maintain this state of, well, essential peace with our own tissues. It's governed by a highly sophisticated, multi-layered regulatory system, and we call that immunologic tolerance.
Okay, so tolerance here isn't just passive acceptance. It sounds much more active, like a regulated process.
Exactly right. Yeah, technically, immunologic tolerance is the phenomenon of unresponsiveness to an antigen, induced by exposure of lymphocytes to that very antigen. Now, when we talk about health and self-preservation, the specific term we really focus on is self-tolerance. That's the critical lack of responsiveness specifically to an individual's own antigens. It's the absolute underlying requirement for our survival. It ensures our defense system doesn't commit, chronic systemic friendly fire.
And the moment that essential truce breaks down, well, the results are devastating. We're talking about the failure of self-tolerance leading directly to autoimmune diseases.
Precisely. The immune system basically misidentifies self-antigens as foreign and, boom, launches an attack. And we see a whole spectrum of these diseases. At the organ-specific end, you've got diseases like type 1 diabetes. Here, autoreactive T cells and antibodies just meticulously destroy the insulin-producing beta cells in the pancreas.
Right.
Or think about multiple sclerosis, where T cells attack the myelin sheath that surrounds nerve fibers in the central nervous system.
And then there are the truly catastrophic systemic diseases, where it seems like the body is attacking antigens just everywhere.
Yeah, the classic example there is systemic lupus erythematosus, SLE. In SLE, the immune response generates this complex, diverse array of antibodies directed against widespread cellular components, things like DNA, histones, protein phospholipid complexes. And this leads to lesions across tons of organs, from the skin right through the kidneys. The scale of this problem is huge. Autoimmunity is estimated to affect, 5%, maybe 10% of the U.S. population? And concerningly, its incidence seems to be rising globally.
Wow. So, okay, our mission today is to dive deep into the strategic blueprint the body uses to maintain this harmony, this tolerance. We're going to classify these tolerance mechanisms into two main categories, right? Two interconnected categories, representing two distinct theaters of operation: central tolerance and peripheral tolerance.
That's it. These two pillars really represent the body's full strategy. You can think of central tolerance as the rigorous quality control during development, making sure dangerous cells are purged before they're even deployed.
The boot camp.
Exactly, the boot camp. And then peripheral tolerance is more like the continuous patrol and peacekeeping force, managing the self-reactive escapees out in the real world in the tissues.
Okay, let's start where the cells are born and trained then. Central tolerance. This sounds like the immune system's highly selective, maybe even brutal boot camp.
It really is the first and, frankly, most brutal checkpoint. Central tolerance happens exclusively during the maturation of lymphocytes in what we call the primary lymphoid organs. So for T cells, that's the thymus. For the B cells, it's the bone marrow. This is the stage where these cells randomly assemble their antigen receptors, TCRs for T cells and BCRs for B cells.
And given that this assembly process is random, there must be a very high statistical probability that a lot of these newly minted receptors will have a dangerous affinity for self-antigens, right? They could potentially attack our own body.
Oh, inevitably. Absolutely. Right. Central tolerance is the mechanism, the brutal mechanism, designed specifically to eliminate these dangerous clones before they ever leave the protective environment of those lymphoid organs.
Okay, let's start with T cells in the thymus. If a developing T cell recognizes a self-antigen, say, displayed by a local antigen-presenting cell in the thymus, what happens?
That cell is immediately targeted for negative selection. Another term for it is clonal deletion. Basically, recognizing a self-antigen is seen as a major failure of quality control. The cell gets signals that trigger rapid, programmed cell death apoptosis.
Ah, apoptosis. Cell suicide.
Exactly. This is the main purging mechanism, and it eliminates the vast majority of potentially dangerous self-reactive T cells.
But here's the thing I'm wrestling with, the big logistical challenge. How does the thymus, which is, sitting there in the center of the chest, how does it know what the body's entire set of self-antigens looks like? It needs to check these T cells against proteins that are only naturally found way out in organs like the liver or the gut or even the central nervous system. That seems like a massive geography problem.
It is the ultimate geography problem. And the solution is, well, arguably one of the most fascinating aspects of tolerance. The body uses a special protein called AR. That stands for autoimmune regulator. You can think of it as a kind of molecular spy master within the thymus.
AIR, a spy master. Okay, tell us more about AIR. How does it bridge that huge geographic gap?
Well, AIR is a type of protein called a transcription factor. And what it does is it stimulates the epithelial cells and the dendritic cells within the thymus to express literally thousands of peripheral tissue-restricted self-antigens.
So antigens normally only found elsewhere.
Exactly. Think of it as forcing the thymus to run this really comprehensive training scenario. The thymic cells suddenly start producing proteins normally only found, say, in the pancreas like insulin itself, or maybe proteins from the thyroid gland or neurological tissue.
Wow. So the immature T cells, while they're still confined to this boot camp, they get exposed to antigens from pretty much every corner of the body. And if they react aggressively to any of these spy antigens presented locally, bam, they're immediately flagged for deletion.
Precisely. It ensures that the T cell repertoire, the whole library of potential attackers, is trained against self-antigens it will encounter everywhere, not just the ones typically found in the lymphoid system itself. It creates this artificially diverse environment specifically to ensure almost nothing slips through.
And the absolute necessity of this AIR safety net is really proven when it fails, isn't it? What are the clinical consequences if someone has a defective AIR gene?
Oh, the clinical picture is stark. It's really telling. Loss-of-function mutations in the AIR gene cause a rare but truly devastating human autoimmune disorder. It's known as the autoimmune polyendocrine syndrome, or APS1.
Polyendocrine,
Yeah.
meaning multiple glands.
Yes. The T cells that were never properly checked against these peripheral antigens manage to escape the thymus, and they then systematically attack multiple endocrine organs. The parathyroid glands, the adrenal glands, the pancreas. It's definitive, really tragic proof that if you compromise this central tolerance mechanism, the immune system just runs rampant.
So deletion, clonal deletion, is the primary fate for these self-reactive T cells. But the source material notes there's this crucial fork in the road, particularly for some of the CD4 plus T cells that recognize self-antigen. They don't die, they become something else instead, a peacekeeping force.
Yes, exactly. Instead of undergoing apoptosis, a specific subset of these self-recognizing CD4 plus T cells is actually instructed to develop into regulatory T cells, or TREGs, as they're often called. The body essentially converts a potential threat into a critical tool for maintaining peace later on out in the periphery.
What determines that choice then? Death or conversion into a TREG?
Yeah, the precise molecular trigger isn't completely nailed down yet, but the working hypothesis relates to the affinity or the strength of the receptor binding. It seems that high affinity recognition of a self-antigen, a really strong reaction, often signals lethal deletion. Whereas maybe slightly lower affinity recognition, a more moderate rather than aggressive reaction, might actually favor the differentiation into a regulatory T cell. The body is essentially saying, this cell is way too dangerous to keep around, let's delete it. But that cell, maybe it can be repurposed to help suppress others.
Fascinating. Okay, now let's switch gears slightly and move to the B cells. They mature in the bone marrow. Do they face the exact same lethal ultimatum as the T cells?
They follow a very similar principle, yes, but they have this really fascinating, unique mechanism that essentially allows them a do-over. This is where we encounter something called receptor editing.
Wait a minute. A cell that recognized a self-antigen is given a second chance to re-engineer its weapons. Doesn't that seem like a massive gamble? Why would the body be willing to take that risk instead of just, deleting the cell like it does with T cells?
It's an energy-intensive gamble, that's true, but it seems to be quite efficient overall. B cells are valuable. They're the ones that generate antibodies. So if a developing B cell strongly recognizes a self-antigen within the bone marrow, the cell actually reactivates the gene rearrangement machinery, the RNEG genes that originally assembled its B cell receptor in the first place.
So it goes back to the drawing board.
It essentially goes back to the drawing board, exactly, and tries to express an entirely new antigen receptor, hoping, of course, that the new receptor will not be self-specific.
It's like hitting the reset button on its weaponry. And this process isn't just some minor niche mechanism either. The sources indicate that anywhere from, what, 1/4 to maybe even 1/2 of all the mature B cells in our body may have undergone receptor editing during their maturation. That's a staggering amount of self-correction happening.
It really underscores the sheer volume of potentially self-reactive receptors that are generated just by chance during that random assembly process. The body seems to prefer retooling the weapon rather than deleting the entire soldier, if you will. However, and this is important, if the editing fails, if the B cell generates yet another self-reactive receptor, then the fail-safe of apoptosis kicks in. Central tolerance ensures that any cell with aggressive self-specificity is dealt with one way or another, whether by retooling or destruction, before it ever gets out into the circulation.
Okay, so central tolerance is this incredibly rigorous screening process, the boot camp. But we established early on that it's inherently imperfect. Not all self-antigens are going to be expressed in the thymus, even with air, and some self-reactive T cells inevitably slip past, reaching the peripheral tissues.
Yeah, that's the critical transition point. That's where we move to peripheral tolerance. This is the second pillar. It's the continuous policing out on the outskirts in the tissues, designed specifically to silence or delete those self-reactive escapees that somehow made it through the central boot camp.
Right. And perhaps the most established mechanism for dealing with these escapees once they're out in the periphery is essentially rendering them functionally paralyzed.
Yes, we call that state energy. Now, to really understand energy, we have to recall the strict rules of T-cell activation, specifically the requirement for two distinct signals.
Okay, this is really fundamental, isn't it? We need to make this crystal clear. What are the two signals required for a T cell to actually launch an attack?
Right. So signal one is the recognition of the antigen itself. This happens when the T-cell receptor, the TCR, binds to a peptide fragment that's being presented by an MHC molecule, that's the major histocompatibility complex, on the surface of an APC, an antigen-presenting cell, like a dendritic cell, for instance. That's signal one, antigen recognition.
Got it. Signal one, see the target. And signal two.
Signal 2 is the crucial costimulatory signal. You can think of it like the required security clearance code, or maybe the second key needed to launch. This is typically provided when molecules like B701 and B702 on the APC bind to the CD28 molecule on the surface of the T cell. You absolutely must have both of these signals to initiate a full T cell response. No second signal, no activation. Okay. And the brilliance, the elegance of energy hinges on the fact that normal resting antigen-presenting cells, the ones just hanging out in tissues, which might inadvertently display a self-antigen, they don't typically express those crucial B7 co-stimulators, do they?
That's the elegant security measure built into the system.
Exactly.
So if an escapee T cell, one that's reactive to self, recognizes a self-antigen, that's signal one, being displayed by just a resting APC without adequate levels of those costimulatory B7 molecules, so lacking signal two, the T cell is rendered functionally unresponsive. It enters this anergic state. It's essentially paralyzed then. It can see the self-antigen, but it just cannot act. And what's really critical here, I think, is that once a T cell becomes anergic, it seems it can't be activated later on, even if a second signal eventually shows up.
That seems to be the case, yes. That's the permanence, or at least the long-lasting nature of the paralysis.
Yeah.
It's like a traffic cop saying, nope, no second signal, no green light for you, and by the way, now you're banned from using this road forever. This mechanism is vital for preventing just a low-level, everyday presentation of self-antigens from constantly triggering unnecessary low-grade inflammation throughout the body.
Okay, that makes sense. But what if we have a really aggressive escapee T cell with high self-reactivity? Maybe just paralysis, energy isn't enough. We might need active suppression, which brings us back nicely to those repurposed peacekeepers you mentioned earlier, the regulatory T cells.
The regulatory T cells, TREGs, yes, they are arguably the most essential players in maintaining peripheral tolerance. They are the active suppressors, the peacekeepers. And they're defined by some specific markers. They are CD4 plus T cells, and they express high levels of the IL-2 receptor alpha chain, that's CD25, and most critically, this master transcription factor called FOXP3.
FOXP3, that seems to be the absolute linchpin, doesn't it? The defining feature that really converts a regular T cell into a dedicated suppressor.
It really is the master regulator for TREG development and function. Both IL-2, which is a cytokine vital for T cell growth and survival in general, and FOXP3 are absolutely required for the development, the survival, and the ongoing function of these TREGs. If you don't have enough functional FOXP3, you simply don't have proper brakes on the immune system.
And once again, like with error, we have a catastrophic human disease state tied directly to the failure of this specific gene.
Absolutely. Mutations in the FOXP3 gene in humans lead to a condition called IPEX syndrome. That stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. It's a devastating, severe systemic autoimmune disease that just hits multiple organ systems simultaneously. It proves beyond any doubt how vital these FOXP3 expressing tregs are for keeping the peace in the periphery. And similarly, even subtle variations or polymorphisms in the gene for CD25, the IL-2 receptor component, are associated with an increased risk of complex autoimmune diseases like type 1 diabetes and multiple sclerosis.
So we have this dedicated peacekeeping force, the tregs. How exactly do they actively suppress their neighbors, the potentially aggressive lymphocytes? Is it like physical competition or more like chemical warfare?
It seems to be a bit of both, actually. Their inhibitory activity is achieved through several different mechanisms. They can certainly compete with other T cells for the limited available supply of IL-2, essentially acting like a sponge and starving out the potential aggressors that need IL-2 to proliferate. But perhaps their primary method is active chemical suppression. They secrete potent immunosuppressive cytokines, chemicals that dampen the immune response. Chief among these are IL-10 and TGF-A, transforming growth factor beta.
Right. IL-10 and TGF-beta, they effectively act like local fire extinguishers, muzzling the local immune response.
They really do. They actively inhibit the activation, the proliferation, and the effector functions of nearby lymphocytes, and even the antigen-presenting cells like dendritic cells. So TREGs don't just stop an ongoing attack, they actively work to restore local tolerance and shut things down.
The source material also gives TREGs a much broader role, almost a global one, beyond just dealing with self-tolerance. It mentions two critical biological scenarios involving foreign antigens that we absolutely must tolerate.
That's a really vital point, yes. First, TREGs are absolutely essential for tolerating the fetus during pregnancy. A developing fetus expresses paternal antigens, which are technically foreign to the mother's immune system. TREGs are crucial in preventing the maternal immune system from recognizing the fetus as foreign and launching an attack against this allograft. It's actually highly suggestive that the evolution of placentation itself, which requires this fetal tolerance, occurred pretty much simultaneously with the ability of the immune system to stably express the FOXP3 transcription factor and generate TREGs.
Wow, that's a deep connection. And the second scenario, our relationship with the trillions upon trillions of commensal microbes living in our gut.
Exactly. These bacteria are technically foreign, they're not self, but TREGs actively suppress immune responses against them, maintaining a necessary truce with our gut flora. This prevents chronic inflammation and damage in the gut lining. So in many ways, TREGs are the ultimate mediators of biological peace, managing tolerance to self, tolerance to the fetus, and tolerance to our essential microbial partners.
Okay, so we've covered paralysis through energy and active suppression by TREGs. But what if all those peripheral mechanisms fail? What about the truly aggressive, self-reactive clones that somehow escape central tolerance and manage to ignore peripheral regulation by energy or TREGs? The body must need a final, decisive option, a final kill switch for these dangerous cells.
Yes, that's the final deletion mechanism operating out in the periphery, inducing apoptosis in these problematic self-reactive cells. The body seems to utilize two main pathways to execute these cells once they're out in the tissues. One involves triggering the internal mitochondrial pathway of apoptosis.
No, okay, this involves that protein called BIM, which sounds a bit technical. How can we simplify that?
Right, let's use the trigger metaphor again. T cells that repeatedly recognize self-antigens out in the periphery might start expressing high levels of a pro-apoptotic protein called BIM. Think of BIM as the internal trust for the cell's self-destruct sequence. Now, normally the cell has safety catches engaged. These are anti-apoptotic proteins, like BCL2 and BCLX, that keep BIM locked down and prevent accidental self-destruction.
Okay, so safety catches are on. But if the T cell keeps seeing self-antigen aggressively,
The persistent signal seems to cause the cell to stop producing those critical anti-apoptotic safety catches, BCL2 and BCLX. Suddenly, BIM is released, it pulls the trigger unopposed, and that initiates the mitochondrial pathway of cell death. Essentially, the aggressive T cell deletes itself through this internal mechanism.
got it. Self-deletion via BIM. And the second mechanism, this involves an external signal, the death receptor pathway.
Correct. This is the fast ligand system. Recognition of chronic self-antigen exposure, especially under inflammatory conditions, can induce lymphocytes to co-express both the death receptor FAS, which is also known as CD95, and its corresponding ligand FAS-L. When Fas-L on one cell binds to Fas on another, or even on the same cell, it triggers the death receptor pathway, leading directly to apoptosis. It's like a targeted assassination signal exchange between cells.
And yet again, we see the clear clinical consequence when this specific kill switch fails. Mutations in either Fas or Fas-L lead to what condition?
They result in autoimmune lymphoproliferative syndrome, or ALPS. This is a condition characterized by abnormally high levels of lymphocytes that should have been deleted, leading to enlarged lymph nodes and spleen, and widespread autoimmunity that often mimics systemic lupus erythematosus, SLE. It clearly shows that the failure of this crucial peripheral cleanup system allows these aggressive, self-reactive cells to survive, proliferate, and cause disease.
Okay. Before we move fully into the clinical implications and breakthroughs, there is one last, maybe more passive, mechanism of tolerance the immune system seems to utilize, simply being unaware that certain self-antigens even exist.
Ah, yes. This is the concept of immune-privileged sites. These are specific tissues in the body that are physically sequestered or shielded from the main highways of the immune system. They're protected by unique anatomical barriers, meaning they don't actively communicate with the lymphatic drainage or the main blood supply in the usual way. Essentially, the antigens within these sites are ignored because the immune system, for the most part, never even sees them.
And the sources cite classic examples like the testes, the eye, particularly the anterior chamber, and parts of the central nervous system, the brain.
Exactly. These are typically sites where any kind of inflammation would be devastatingly destructive to function. So the tolerance here relies primarily on physical ignorance or sequestration. However, and this is the important caveat, if these sequestered antigens are suddenly released into the general circulation, perhaps due to severe trauma, like a penetrating eye injury or testicular damage, the body's immune system, which has never been trained to tolerate them centrally or peripherally, may suddenly see them as foreign and launch an aggressive immune response. This can potentially lead to destructive inflammatory conditions like post-traumatic uveitis in the eye or orchitis in the testes.
Right, the ignorance is broken and the attack begins.
Okay, we spent a good chunk of time detailing the defensive side of the equation. All these intricate mechanisms designed to establish and maintain tolerance. Now, let's pivot to the really exciting clinical frontier, because understanding these very mechanisms has unlocked massive breakthroughs, particularly in cancer therapy.
Yes, this brings us squarely to the molecular breaks of the immune system. We're talking about the inhibitory receptors, specifically CTLA-4 and PD-1. Now, interestingly, they're structurally related to that activating CD28 molecule we discussed earlier, the one needed for signal 2. But CTLA-4 and PD-1 serve the completely opposite function. They act as brakes, they dampen the immune response. They are absolutely crucial components of peripheral tolerance designed to shut things down.
Okay, so CTLA-4 acts as a functional brake primarily by binding to those same B7 molecules, the very ligands that CD28 needs for T-cell activation. But CTLA-4 binds them with a much higher affinity, right?
Yeah.
Exactly. It essentially acts as an off switch by outcompeting CD28 and stealing that necessary co-stimulation signal away. PD-1, which stands for programmed death 1, also acts as a crucial brake. When it binds to its ligands, primarily PDL1 and PDL2, which can be expressed on various cells, including tumor cells, it essentially sends a signal telling the T-cell, stand down, stop attacking, tolerate this.
And the clinical significance, the importance of these breaks is again highlighted by observing what happens when they fail genetically.
Precisely. Just like we saw with FOXP3 in AIR, inherited mutations in the CTLA-4 gene in humans result in severe systemic inflammatory diseases.
Yeah.
If you lose this crucial brake, the T-cells just accelerate uncontrollably. So these receptors exist specifically to ensure that once an immune attack on a foreign invader, like a virus, is successful, the response is rapidly shut down to prevent excessive inflammation and collateral damage to self-tissues.
But here is the profound clinical paradox, the insight that really led to Nobel Prize-winning research. These very breaks, which are designed to protect us from ourselves by enforcing tolerance, are cleverly exploited by our deadliest internal enemies, cancers.
That's exactly right. Malignant tumors and also some persistent viruses figured out that they could evade the body's powerful immune defenses by simply turning those brakes on. Many types of cancer cells, for instance, express high levels of PDL1 on their surface. So when an anti-tumor T-cell approaches, ready to attack, the tumor cell essentially shakes the hand of the T-cell using its PDL1, engaging the T-cell's PD-1 brake. This forces the T-cell into a tolerated, exhausted, or anergic state, effectively neutralizing the attack.
Wow. So the body has this powerful defense weapon, the T cell, but the tumor cleverly exploits the self-tolerance mechanism, the PD-1 pathway, to put the brakes on and protect itself.
That realization was the absolute breakthrough. It led directly to the development of what we now call checkpoint blockade immunotherapy. We developed highly specific blocking antibodies, these are drugs, that physically target and prevent the engagement of these inhibitory pathways. We have anti-CTLA-4 antibodies
Yes.
and anti-PD-1 or anti-PD-L1 antibodies.
And by administering these antibodies to a cancer patient, we effectively remove the brakes on their T cells.
We unleash the full killing potential of the patient's own immune system. We promote a robust, aggressive anti-tumor response that allows the T cells to finally recognize and destroy the cancerous cells they were previously being forced to tolerate. It has been one of the most successful and revolutionary advances on oncology in decades, but it's fundamentally a direct manipulation, a deliberate overriding of the sophisticated tolerance systems we've been detailing.
This incredible success in manipulating tolerance for therapy brings us naturally to the last major area for our deep dive today. How does tolerance ultimately fail in the first place, leading to spontaneous autoimmunity? It's clearly not just one single thing going wrong, is it?
No, almost never. Autoimmunity is almost always multifactorial. The general consensus, which is well represented in the source material under the pathogenesis of autoimmunity, is that it typically requires at least two major ingredients to align. First, the inheritance of certain predisposing susceptibility genes, and second, the presence of the right kind of environmental trigger, very often an infection or possibly tissue injury.
Okay, let's start with the genes. We know that the HLA alleles, those genes that encode the MHC molecules responsible for presenting antigens, are very strongly associated with susceptibility to nearly every autoimmune disease.
Oh, the association is absolutely undeniable and often very striking. For instance, the chance of developing ankylosing spondylitis, a type of inflammatory arthritis, is something like 100 to 200 times greater if you happen to carry the HLA-B27 allele compared to someone who doesn't. Now, the exact mechanism is complex and likely varies between diseases, but a leading theory is that certain HLA molecules are simply better, or perhaps worse, at displaying specific self-peptides to T cells. This might lead to presenting the wrong signal to T cells out in the periphery, or perhaps failing to properly educate T cells during central tolerance in the thymus.
But the genetic landscape for autoimmunity goes much deeper than just HLA, right? The sources mention other non-MHC genes involved in immune regulation, like one called the PTPN22 gene.
Yes, PTPN22 is a great example of a more subtle genetic vulnerability. This gene encodes an enzyme called a protein tyrosine phosphatase. These phosphatases are critical regulators. Their job is to remove phosphate groups from other proteins, and in lymphocytes, this essentially acts as an off switch, dampening down the signaling pathways that lead to activation.
So it's another critical mechanism for maintaining that necessary quiet state, that balance required for tolerance.
Exactly. And it turns out that certain common polymorphisms or variations in the PTPN22 gene are strongly associated with an increased risk of developing type 1 diabetes, rheumatoid arthritis, Graves' disease, and several other autoimmune disorders. It's hypothesized that the disease-associated variants of this phosphatase are functionally defective, perhaps hyperactive or hypoactive depending on the context. They maybe can't adequately dampen down lymphocyte signaling, meaning the cells might be constantly on a higher alert status than they should be, violating that fine balance needed for self-tolerance. The net result seems to be hyperactivity in the immune response pathway.
And crucially, overlaying these susceptibility genes, we have those rare but highly informative single gene mutations we've been discussing all along, the knockouts that directly affect the core tolerance machinery itself. Mutations in AIR, CTLA4, FOXP3, and the apoptosis genes FAS and FASL.
Exactly. These single gene failures, while individually rare, are absolutely essential for our understanding because they provide irrefutable causal evidence. They show that compromising any single major component of either the central or the peripheral tolerance machinery is, by itself, sufficient to cause severe, often systemic autoimmune disease in humans. They are the definitive proof of concept for the importance of the whole system.
Okay, so you have the genetic susceptibility, the potentially faulty hardware. What then typically lights the fuse? You mentioned environmental triggers, specifically infections.
Yes, infections are strongly implicated as triggers that can break tolerance in genetically susceptible individuals.
Yeah.
There are two primary postulated mechanisms for how this might happen. The first is often called nonspecific bystander activation.
Bystander activation.
Yeah. Imagine a microbe, like a virus or maybe significant cellular injury from trauma, initiates a strong local inflammatory reaction in a tissue. This inflammation forces the local antigen-presenting cells, those dendritic cells, to mature and express high levels of those crucial costimulators, B7-1 and B7-2, on their surfaces. Remember, resting APCs usually don't express much B7.
Ah, so we're back to overcoming energy.
Precisely. Now, imagine a self-reactive T cell that was previously harmlessly antinergic in that tissue because it only got signal 1, self-antigen, but lacks signal 2, co-stimulation. Suddenly, it encounters its self-antigen being presented by an APC that is now highly activated by the nearby infection or inflammation and is therefore expressing lots of B7. That T cell now receives both signals, self-antigen plus co-stimulation, and boom, it's fully activated against the self. The infection essentially provided the necessary danger signal to overcome the T cell's previously established functional paralysis.
Okay, that makes sense. The infection wakes up the APCs, which then accidentally activate nearby self-reactive cells. What's the second mechanism? Something more insidious. Molecular mimicry.
Right. Molecular mimicry occurs when some microbes happen to express antigens, proteins, or peptides that share amino acid sequences or structural similarities with specific self-antigens found in our own tissues. The immune response, which is legitimately and aggressively targeting the microbial antigen during the infection, might then cross-react and mistakenly activate self-reactive lymphocytes simply because the self-antigen looks structurally similar enough to the pathogen's antigen.
And the classic, often cited example of this in textbooks is rheumatic heart disease, isn't it?
That's the canonical example, yes. Following certain streptococcal infections, like strep throat, antibodies generated against specific streptococcal proteins can unfortunately cross-react with structurally similar proteins found in human heart muscle and heart valves. This cross-reaction causes inflammation and damage, leading to myocarditis and valvulitis. While rheumatic fever is becoming less common with antibiotic use, this mimicry mechanism is still suspected to play a more subtle, perhaps contributing role in the initiation or perpetuation of many other chronic autoimmune conditions.
And finally, just touching briefly on it, the sources also list abnormalities in the way self-antigens are displayed or handled, with SLE, lupus being the major example here.
Yes, in SLE, it's thought that various factors, perhaps including genetic susceptibility and environmental triggers like exposure to UV light, can lead to increased rates of cell death, apoptosis. Furthermore, there might be defects in the efficient clearance of these apoptotic cells by phagocytes. The result is an abnormally high burden of nuclear antigens, things like DNA, histones, and other nuclear proteins being exposed in the tissues.
And when you have those escapee B cells and T cells lurking around, the ones that happen to react to these self-nuclear antigens, they become strongly stimulated, initiating a devastating positive feedback cycle.
Exactly. And this is where the innate immune system seems to get improperly engaged as well. The complexes formed between the nuclear self-antigen and the autoantibodies produced by B cells combine to other B cells and also to dendritic cells. Once internalized, the nucleic acid components, the DNA or RNA, of the self-antigen engage specialized innate immune receptors called toll-like receptors, TLRs, specifically TLR7 and 9, which normally function to sense microbial nucleic acids. So in SLE, the body's own DNA and RNA effectively mimic their microbial counterparts, triggering these TLRs, which powerfully amplifies the immune response uncontrollably, leading to the production of more autoantibodies, type I interferon production, chronic inflammation, and widespread tissue damage.
Wow. This whole process really underscores that autoimmunity is rarely the result of just a single catastrophic failure, but much more often a kind of perfect storm where defective tolerance mechanisms, perhaps abnormal antigen presentation, and environmental inflammatory triggers all converge to light the fuse in a genetically susceptible person.
Absolutely. The immune system normally operates on this incredibly fine tightrope walk between activation, which we need for defense against pathogens, and tolerance, which we need for self-preservation. Autoimmunity is essentially the result of that delicate equilibrium tipping disastrously and often permanently toward perpetual aggressive activation against self.
So let's try and wrap up this incredibly detailed deep dive by synthesizing everything into maybe a simple summary of the strategic paths, the different fates the body has for dealing with a potentially dangerous self-reactive lymphocyte.
Okay. We basically identified 3 primary fates for these cells, depending largely on where and when they encounter self-antigen. First, for immature cells during central tolerance in the thymus or bone marrow, they're either killed outright via clonal deletion, that's the primary purge, especially for T cells, helped crucially by air presenting those peripheral antigens. Or, specifically for B cells, they might be given a second chance to fix their faulty weapon via receptor editing.
Right.
Mhm.
Kill or retool in the center. And then, once those mature, potentially self-reactive cells escape out into the periphery, the strategy shifts more towards active control and containment. Out there, they are either paralyzed by energy, meaning they see the antigen but are functionally blocked by the lack of that critical co-stimulatory second signal.
Or they are actively suppressed by those dedicated peacekeeping regulatory T cells, using their powerful inhibitory cytokines like IL-10 and TGF-beta.
Or, if paralysis and suppression fail as the final and permanent resort for the most dangerous escapees, they are killed by peripheral apoptosis mechanisms, such as triggering the fast-fizzle death receptor system or the internal BIM pathway.
That's a good summary. Kill, retool, paralyze, suppress, or kill again. Those are the options. And understanding why autoimmune diseases are often so devastatingly chronic and progressive comes back to the immune system's own power. When those powerful amplification loops, designed to fight off pathogens effectively, are mistakenly turned against self-tissues, the initial injury causes damage. That damage, in turn, releases more self-antigens, which then activates more self-reactive lymphocytes. You get this process called epitope spreading, where the immune response keeps generating reactions to new self-targets over time, constantly feeding the fire of inflammation.
It truly is an incredibly high-stakes balancing act, built right into the core of our biology.
Which brings us right back to the biggest clinical implication and maybe the final provocative thought for you, our listener, to ponder. We firmly established that these tolerance mechanisms, energy, TREGs, CTLA-4, PD-1, are the essential brakes on the immune system. And crucially, we have now learned how to manipulate those brakes clinically. We can physically block PD-1 and CTLA-4 to successfully treat certain cancers. We want the immune system to run aggressively against the tumor in that context.
But, as always in biology, this success comes at a cost, doesn't it? There's a fundamental biological trade-off involved.
There absolutely is. By deliberately removing these natural brakes to fight cancer, we necessarily and significantly increase the risk of inducing severe, sometimes life-threatening systemic autoimmune diseases in those patients, the very conditions that result from natural tolerance failure. So the really profound question for the future of medicine, particularly immunotherapy, is this. As we continue to refine and push the boundaries, learning how to better unleash an aggressive, potentially life-saving immune response against a deadly cancer, or maybe even a chronic infection, how will we simultaneously learn to control, predict, and manage the precise degree of systemic autoimmunity we are willing to risk unleashing? We know that tolerance failure can lead to chronic, progressive, and sometimes fatal self-attack. It's going to be a constant, high-stakes clinical trade-off that truly defines the cutting edge of immunology for years to come.